Hello. Hi, everyone. Thank you for joining our MIDD Plus 2023 clinical track program. Apology for a short technical delay. My name is Tom Sun, VP of Strategic Consulting Services at Cardigen Simulation Plus. It is my pleasure to introduce our next speaker, Dr. Naveed Shaik from Gilead. Um, he's currently a senior clinical pharmacologist too at Gilead Sciences. And the talk of his title will be Physiologically Pharmacokinetics Model-Based Approach for Predicting Dose for Low-Acting Lenacapavir. Before the start of presentation, I'd like to remind everybody that we take your privacy rights very seriously by participating our section that you're allowing us to contact you for follow-up. And you may ask any questions for using the Q&A tab on your dashboard anytime. Any technical questions, please type in the chat. We will try to help you. And without further ado, Naveed, please start your presentation. Thank you very much.
Sure. Apologies for the technical issue. Thanks, Tom, for the nice introduction. Good morning and good afternoon, everyone. I am Naveed Shaik. As Tom mentioned, I'm from Gilead Sciences, one of the quantitative pharmacologists or clinical pharmacologists on lenacapavir program. I have been working on this program, and I'll be presenting a PBPK-based modeling approach, what we have used for lenacapavir model development and translation of the models from preclinical to clinical, as well as predicting the concentrations based on the knowledge gained from the different species. So having said that, just to give an outline of my talk, I'll go through the background about lenacapavir, its mechanism of action, and how it is being dosed, the dosing nomogram for lenacapavir, and we'll jump into the PBPK model, what we are trying to achieve through this PBPK model, as well as some of the conclusions so far. It's a work in progress, so the conclusions are up to what we have achieved so far. Sorry. Lenacapavir is a first-in-class capsid inhibitor of HIV. It has been approved for the treatment of HIV. Um, with other antiretrovirals, especially in treatment experience living with HIV. Um, on top of it, it is also being developed as a prevention for HIV. Uh, as well. So in the ongoing phase 2, 3 studies with people living with HIV, we dose lenacapavir sub-Q 927 milligram for a 6-month dosing, but before that we give them a oral lead-in period where the oral drug is given on days 1 and 2 with two tablets of 300 milligram and one tablet of 300 milligram on day 8, which will increase the concentrations and attain the concentrations required. So once it is there, the sub-Q is given on day 15, which will have a sustained release over a period of few months, and it will maintain the concentrations required for the duration of the injection. Um, Lenacapavir has a very characteristics suitable for the long-acting development, long-acting drug development. So it exhibits very low solubility and permeability, which makes it a very suitable candidate for developing as a long-acting injectable. Once given as a sub-Q injection, the len is slowly absorbed over a period of few months, and the maximum concentrations are achieved around 77 to 84 days, and the apparent half-life is around 10 to 12 weeks for the sub-Q administration. This is because we we have a absorption limitation happening from the sub-Q depot. So you, we have different formulations being worked, and the current formulation which has been approved is a simple lenacapavir result with PEG 300 and water, nothing else, which will make it a long-acting or a controlled release compound. In vitro, as well as the non-clinical and clinical data has suggested that the len, once given as a sub-Q injection, due to its low aqueous solubility, it forms a depot. Once the excipients, the PEG and the water, once they are absorbed, the the depot is slowly absorbed or dissolved, and enters into the systemic circulation over a period of a few months, maintaining the concentrations required for LEN to bring its activity. Having said that, mechanism of action of lenacapavir, it is a multi-stage inhibitor of HIV-1 capsid, and the red highlighted areas are different stages where lenacapavir acts on the HIV-1. So there are three different areas, especially the capsid nuclear transport, the virus production, and the capsid assembly, acting with a very strong EC50. The proof of concept studies, where we have injected the lenacapavir subcutaneously with different doses ranging from 20 milligram to 750 milligram. Uh, we we have seen a 1.3 log, at least a 1.3 log reduction in HIV RNA on day 10, based on the concentrations what we have seen on day 10. So this has established the proof of concept that lenacapavir will reduce the HIV, and at least, as we have seen here, 1.3 log units on day 10. One of the challenges what why we have are looking into the BBK modeling of lenacapavir with the long-acting injectables, especially with lenacapavir, which is given every 6 months. So the clinical development is kind of like prolonged. One of the shortcuts or areas which can be improved the drug development time is by developing the modeling approaches, and especially PBPK modeling, where for the long act, if we can utilize that one, it can save the time and resources. So uh, lenacapavir demonstrates flip-flop kinetics. It's absorption limited. The absorption is the rate limiting step over there. And then once we can capture the PK profile of the sub-Q between different species, especially from the non-clinical species where we have data, and use the same characteristics of release in the humans, then it can be applied, and the time can be reduced to predict the concentrations for a particular formulation. So why we have chosen PVPK, especially the physiology of is being considered over here, how the drug enters and forms a depot, and how the tissue is being acted upon by, and the drug slowly releases. That's the reason why physiology comes very handy in predicting the drug concentrations, the absorption profile of the lenacapavir. So with the PVPK modeling approach for lenacapavir, we wanted to describe the characteristics of len non-clinical as well as the clinical PK, develop the PVPK model for the subcutaneous routes with different formulations, and each particular formulation establish a translation process where the release kinetics for one particular formulation can be translated from a non-clinical species into the clinical data. So with that, approach or our objective was to simulate multiple different formulations as well as look into different routes of administration, not only the subcutaneous administration, but also the IM and at different sites. This is the PVPK model development and methodology. It's very typical where we start with the physiochemical and biopharmaceutical properties. All the in vitro parameters, whatever are available, are taken into development of the model. The parameters which were lacking or were not available, we have tried to predict those parameters, and we were lucky enough to have the IV data not only for clinical species but for the clinical species as well in the clinic as well, which gave us a good handle on developing the PBPK model, adjusting the volume of distribution, clearance, and once we had developed the IV models, we further utilized, expanded it to the oral models as well, keeping the volume of distribution and clearance fixed and working on the absorption processes. Similarly, once that was done, we also worked on the subcutaneous administration, which we have tried multiple different things. Available literature for the long-acting work that has been done with PBPK, as well as looking into the physiologies, how this drug is being absorbed over a period of time. So we started with the non-clinical species. Once we have established the sub-Q models for the non-clinical species for a particular formulation, we tried to mimic that for the same formulation in clinic and establish or get the plasma concentration time profiles correct. Overall, as I have mentioned, the clinical regimen includes a oral lead-in where we have to give the oral drug initially for 2 weeks, and then we continue with the subcutaneous administration of lenacapavir for every 6 months. So our goal is to have different formulation, predict the concentrations from a different formulation, and see if we need an oral lead-in with those formulations. Excuse me. So for for our PBPK modeling work, we have used GastroPlus, and the default physiologies are taking into consideration the body weights and the demographics available from our clinical studies. For the humans as well as the non-clinical studies, body weights and other aspects into consideration when we develop the model. For the sub-Q administration, we had to come up with a workaround where we have the excipients like PEG and water, which get fastly absorbed over few hours, and then the drug is left. Which gets slowly absorbed over a period of time, which I'll show in next couple of slides how we have modeled this one. So the first portion, when we have the excipients, it's a solution and the drug gets fast fastly absorbed, whereas once the excipients are absorbed, the drug remains and the solid particles represent the precipitate. This precipitate forms a depot formation, and this depot is absorbed over a period of time, and which has been fitted with a viable function. So for each of the formulation, what we have with the different excipients, viable function or viable parameters were fitted, and those parameters were for the rat, and were able to capture the drug concentration profile for that particular formulation in humans as well. Having said that, I'll just quickly go over the IV model. It's the typical PBPK model where we are trying to fit the volume of distribution and clearance. The blue dots over here are the observed data in triplicates from the rat. Kg solid line is the simulated one. So with the methodologies which we typically use for PBPK modeling, we were able to capture the data reasonably well, and the PK parameters are given on the right side in the table. We were within the BE bounds for the rat. Similarly, for humans, we had two different dose levels, 10 milligram and 20 milligram IV doses, and we did the same the rat, and we're able to capture the data reasonably well. Excuse me. For the sub-Q administration, we have tried out different methodologies and came up with a methodology where the drug is given or the drug absorption is in a different way, where we have the initial solution formulation because the excipients keep the drug in solution, and then once the excipients are absorbed, all the drug is remained in solid particles, which precipitates and absorbs over a period of time. So for that, we did the mixed multiple dosing, where less than 1% of the drug was given as a solution, and the more than 99% of the drug was being absorbed through a control mechanism, which was defined by viable function. So for different formulations, we had different viable functions, either triple, double viable, triple viable, etc. So once we were able to fit the viable parameters for each of the formulations, we were able to capture the data well for a single dose over here shown for in the rats with a 50 milligram per kilogram dose. The same blue dots are the observed data, and the red line is the simulated one. With a single injection, we can see that the concentrations are maintained up to 52 weeks, which is almost a year over here. And what I have talked about the mixed multiple dose in the previous slide, where we have the initial portion being absorbed as a solution, and that is this shaded area, which is highlighted over here, up to 3 weeks, where we see a small peak and then slowly a sustained absorption over a period of time that will give another actual peak for the entire profile. So once we had this viable parameters established from one of the formulations, the same formulation was used, same viable parameters were used for the same formulation in human data as well. So we had a bunch of formulations, and each formulation were slightly had a slightly changed viable parameters, and it was able to fit the profiles really well. And these are the PK parameters, especially the Cmax and AUC, and as we can see, they were very reasonably well captured within the BE limits. For formulation, where we had established the viable parameters in the preclinical species, we were able to fit that to the same formulation in humans and capture the data reasonably well. So over here we had three different doses, or two different dose strengths with the same formulation. One is 309 mg injection, 309 with a 1 ml injection, and the others are 927 milligram injection, either given as a 1 ml injections or 1.5 ml injections. Again, we use the same approach, solution versus the solid particle approach, and the solution approach, as we can see over here. With the insert, we were able to capture the first initial peak, and then after that there was a slow sustained release, as can be seen with all these three different profiles. So with that, sorry, again, the parameters were captured within the BE bounds, and the AUC and Cmax were either within 0.93 to 1.19 fold, or 1.08 to 1.23 fold. And so with this approach, where we had two different doses, or a dose divided into two different routes. One is the solution, and the other one solid particle. We were able to capture the data from the preclinical and translate it to the humans. So far, based on the work what we have done uh, with the formulations, both in non-clinical and the clinical, PBPK model was developed with the mixed multiple dosing, and we were able to capture that with the solid particle versus the solution fraction, and describe the PK profiles really well across multiple dose levels, and also across the formulations, including the SC administration and IV. So what we are trying to do is, like, once we have this proof of concept which is established with one of the formulation, we can use the other formulations for preclinical and predict the human concentrations, and try to overcome any of the potential, like, long studies. With that, this is our team, the ClinFarm team. Vamsi is the lead for one of the phase 2-3 study. Renu is the overall ClinFarm lead. Ramesh is the TA. We are overseen by Sandhya. Um, we have our clinical research folks with whom we work closely, and we have also collaborated with Viera, which has significantly improved on the PBPK modeling aspect, what we have developed. Any questions? Happy to take on.
Thank you, Naveed. Very insightful presentation. I really appreciate, you really keep your commitment to join our conference when you are not feeling well, and really appreciate that you join us for this present. There's one question from the audience that how do you mechanistically justify using a triple or double Weibull function to describe the drug release from the depot? So could you briefly address that? Thank you.
Sure. So the triple or the double has been fitted based on what we have data into the preclinical, and fitting, as we can see over here in one of these profiles. So any changes in the viable functions can lead to either changes in the tail end or in the middle portion. So to capture that, we had to fit the viable functions and match the profiles.
Great. Thank you for the answer.
No problem.
It's 1:20 now. We need to end this session. I want to thank you again for your presentation, and audience for joining us. Sure. We do have a Q&A sections at 10 after 2 o'clock Eastern time, US time zone. So please rejoin us for additional questions for Naveed and other speakers in this clinical sections. So want to thank you again for your presentation, everyone, to join us.
Thank you.
We're gonna end this broadcast. Thank you very much.
Thank you, everyone. Thanks, Tom.